Good morning. I'm Michael Greenwood in the HLA lab. And with us today is Dr. Driscoll. She's studied physics and got her PhD from University of Maryland before moving to Texas to work at UT Southwestern, and then moving up here to work at the U. So she's giving a talk today, Digging Tunnels, Shaping Signaling, and Other Functions of Cellular Morphology. Dr. Driscoll, thanks for coming.
Well, thank you. Yeah, I don't normally get to talk to not basic science department, so it's exciting to share my work today. And I just started my lab here in the summer, last summer, so we're just getting going. Okay, all right, so these are 3 individual cells. And cells in your body have intricate and convoluted morphologies, right? This is a dendritic cell. It's a type of immune cell on the left. There's a melanoma cell in the middle. And this is a model for a lung cancer cell on the right. And we know that cell shape is indicative of cell state. I think that's where the most famous example of that probably comes from pathology. It comes from diagnosing cancer from pathology slides. But cell morphology also directly facilitates cell functions. Okay. Yeah. Right. So we know that the shapes of neurons help allow us to think. Right. Cilia in the lungs help us to breathe. And furthermore, not only cell shape, but the dynamics of cell shape also facilitate cell functions, right? So these are, it's hard to image in vivo, but this is, these are metastasizing cancer cells in a mouse. And you can see that these cells are extremely dynamic. The nuclei are in green. And the cell shape is in red, although it's hard to see. But they're moving, not only is the boundary, the overall boundary of these cells moving, but the cells are moving within the group as well, right? And it turns out that I actually come from, I went to grad school intending to study dynamics in the abstract, not intending to study biology at all. So I'm very interested in how dynamics is created across scales. And it turns out that the dynamics of cells it is created from sort of cell dynamics is regulated by dynamics across scales, right? So physically, cell shape is created by forces at the cell membrane. Cells create their own forces internally by growing and controlling a physical thing, the cytoskeleton, right? But this physical cytoskeleton, here we see the assembly of an actin network, is in turn controlled by localized intracellular signaling. These are, again, individual cells. We see that actin, these are actin regulators. We see that the cytoskeleton is regulated at the micron scale and below, right? So cell dynamics, it is regulated, is controlled via the cytoskeleton, which is regulated via various intracellular signaling pathways. And to complicate things even further, we know that cell shape itself can alter intracellular signaling, right? So this is a very recent paper. I'm not going to talk about it, but it uses the same cells and the same systems that I am going to talk about. So if you're interested in looking up this paper afterwards, it was just published last month, and I would go for it. Right here, we know that septins, which are localized to which are a filament that localizes to the base of blebs, and hence in that way, blebs are these sort of spherical balls protruding from the cells. Septins sense the curvature at the base of these balls and then alter signaling to increase cell survival, right? So these cells survive by changing their shape, which in turn changes their signaling, right? So now we have a somewhat complicated feedback loop, right? Cell morphology is controlled by cytoskeletal dynamics, which is controlled by intracellular signaling very locally, right? Remember at the submicron scale, which is again controlling cell morphology, right? So the system is very complicated. And today I'm going to talk about I'm going to try and focus on a very particular problem, but we'll also think more broadly about how we understand how cell shape is regulated and how that affects cell function. Right, so we've known for a while that these rounded amoeboid melanoma cells are more associated with metastasis than these long branched melanoma cells that we call mesenchymal. Right, there's work in the literature from various groups, and we measured this again by putting melanoma cells from patients in a variety of artificial environments, and we found that across multiple artificial environments, the cells from more metastatic tumors tended to adopt this rounded form. Okay, so we wanted to know why. Why is this form more dangerous? Why are these little rounded cells more dangerous than this branched form? Okay, and an obvious question when it comes to metastasis, right, is migration, right? So are these rounded amoeboid cells capable of migration in very crowded environments? And that's we have a known, the field has an extremely a relatively well explored model for how amoeboid cells migrate. And amoeboid in this case just means it looks like a giant blob. It looks like a sort of amoeba. And the hypothesis is that amoeboid cells migrate through crowded environments like they would find in vivo by changing their shape in order to squeeze through. And we call it sort of like they're snaking through, right? If this hole is too small to get through, the amoeba can squish itself, can make itself thinner there and squish through, right? But the problem with this model for melanoma cells is people have often reported melanoma cells in vivo that don't look like they're being squashed by their environment, but instead look nearly round, right? So this is a drawing from, I don't look at in vivo cells, but this is a drawing of melanoma in vivo, and we see that the rounded amoeboid form of the cells is at the invasive front. This is a very similar type of melanoma cell in a zebrafish, right? We can image within transparent zebrafish much more easily than we can within the human body, right? So we can image the dynamics of melanoma in a zebrafish. And in both cases, the cells are round, which is weird because you wouldn't expect that if a cell moves in the body by changing its shape, that it would be on average round, right? So we put these cells in 3D collagen, and this collagen is very dense. I took out the slide, maybe unfortunately, but these cells cannot possibly get through this collagen without changing their shape because the collagen is so dense, and yet they are getting through the collagen, okay? So how does a large sphere essentially fit through a very small hole? And also, at least in artificial collagen, these cells are moving incredibly slowly. Fast amoeboid migration can move at 20 micrometers a minute. These cells are moving at 0.03 micrometers a minute, right? But they're moving, and they're metastatic melanoma, so however fast they move, we don't want them to move, okay? It's not clear if it matters how quickly they move. They are moving. We have, well, let's go back. We have something of a problem in that it's difficult overall to image. It's difficult to image dynamics in any sort of realistic environment. And it's particularly difficult to image in 3D in any sort of realistic environment. These are pretty low resolution 2D images. Yet we want to study cell shape. We want to know how cell shape is enabling these cells to get through dense environments, right? And if you put these cells, conventional microscopy as it's used in cell biology often studies cells on a flat surface, but if you take a melanoma cell and put it on a flat surface, it will spread itself out on that surface like a water droplet, right? So this is a melanoma cell on a flat surface and will look nothing like how the cell would look in 3D collagen, or even or in vivo, right? So the same type of cell on the right is in 3D collagen. So we don't want to study cells in 2D, even though that's how most of our microscopes are set up to look at them, right? Instead, we want to be able to study cells dynamically in 3D, and the standard microscopy is just incapable of doing this, right? No. And furthermore, morphology, again, affects not only how cells function, it also affects signaling within cells. So this is a model I remade actually from Dave Odie's group. Dave Odie is in the Department of Biomechanical Engineering on this campus. His office, I think, is directly above mine. But here we've simulated a very simple chemical reaction inside a sphere. So it's a simple model of intracellular signaling. And we see just in this sort of toy model that 15% of the molecules are active. And if we take this same reaction and put it inside a fried egg sort of morphology, which is the morphology the cells adapt if you let them fall onto a flat surface, we see that suddenly 50% of the molecules are active, right? So this is just a toy model, but it shows that even in almost the simplest possible signaling reaction we could imagine, the cell morphology affects the outcome of that reaction, right? So we want to study morphology in 3D, not only because it is required to understand how cells function, but also because it signaling in 3D is changed, which could change other aspects of how the cell state, right? So a little over 10 years ago now, microscopists started to develop a new type of microscope called a high resolution light sheet microscope. And in a light sheet microscope, you don't, you generate a sheet of light and then move that sheet of light through the sample, okay? And because you're and then you image perpendicular to the sheet of light, right? And in part because you're lighting up only the area that you're imaging, you have low phototoxicity, and you could generate a 3D sample very quickly, right? You have to play some tricks to get the sheet of light thin enough for this to work. And there are a wide variety of ways of doing that. But once you do that, you can actually begin to image quickly enough to see dynamics in 3D, right? Before we couldn't even see how these cells were changing their dynamics, even in an artificial sample, okay? So early on in my postdoc, we developed a microscope specifically to look at a high resolution light sheet microscope, specifically to look at cells in very soft environments, right? Because these melanoma cells, it turns out, preferentially are rounded in very soft environments. This is a melanoma cell surrounded by a cube of collagen. Right, this is a different type of cell. It's not a melanoma cell with philopodia. You can see that unusually for a fluorescence microscope, these cells are live, they're moving. Our Z resolution is nearly the same as our XY resolution, and that makes it much easier to understand what's going on here. Okay, so now we can image the melanoma cells in a soft environment and try and begin to figure out how they're moving. Right, this is again a melanoma cell. It's forming what are called blebs, these little sort of balloons on the cell surface. The blebs occur when the plasma membrane becomes locally detached from the cytoskeleton, and the membrane balloons outward, really like a balloon. And these blebs are very fast. They expand and retract about every 20 seconds, which if you'll remember is much, much faster than our 0.03 microns per minute speed, right? So these cells are extending and retracting blebs much, much, much faster than they are moving, right? So it's important to remember here that there are dynamics across many different scales. Okay, and it doesn't take long, really, when you put cells in soft collagen to figure out how they are getting through the collagen with these sorts of imaging. Well, with these sorts of microscopes, it doesn't take a lot to guess that the cell is tunneling through the collagen. We actually can't see it. We still can't see it with other types of microscopes. It's remarkably hard to see. Okay, because the way microscopes work, there's a lot of background from the collagen and trying to see a whole, given that there's so much background from the rest of the collagen, it is difficult unless you have very high resolution imaging. I mean, we have seen it in other microscopes, but it is much harder to do. Okay, so this is a single melanoma cell. It's colored by surface curvature. So red are regions of high surface curvature, or blue are regions of negative surface curvature, and then the blebs show up red in this rendering, right? So the next sort of obvious question to ask is, okay, the cells are making giant tunnels in the ECM, but how are they making tunnels? And it turns out that the most common type of migration is mesenchymal migration, where cells proteolytically degrade a path through the extracellular matrix, right? So amoeboid migration is thought to be distinct from mesenchymal migration, but we wondered if the cells were also, if these melanoma cells were, like mesenchymal cells, proteolytically degrading a path, right? And I'm not showing you all the data here, but we inhibited proteases, we inhibited the molecules that sort of chemically tear the ECM apart in a wide variety of ways. We tried different types of collagen. We tried visualizing where the collagen was torn up. And although these cells do use MMPs, although proteases are degrading the matrix at large, proteases appear to not in any way be connected to that tunneling, right? However, we inhibited the proteases, the tunneling looks the same, okay? So then if the cells aren't using MMPs to make that tunnel, if they're not proteolytically degrading a path through, how are they digging such a giant dramatic tunnel? Okay. And to answer that question, I want to step back a bit. It turns out that I've mostly been showing you projections of 3D images rather than the full 3D image. But the images from these microscopes are quite complicated and difficult to make sense of as movies. This is a single dendritic cell in collagen. The dendritic cell is the actin that in the dendritic cell is labeled in red, right? And see that the cell has an incredibly complicated shape. You may not be as, most of the audiences I talk to are amazed by how complex cell shapes would be. Cell shapes can be, you may be less so than other audiences, but nevertheless, it's difficult to make sense of movies like this. But one simple thing you can do is to segment these cells, right? Is to separate the foreground of the cell from the background of everything else. So here I've colored the cell membrane by curvature. So again, red are regions of high curvature and blue are regions of negative curvature. And then we can see the collagen filling in around the cell. This gives us a better idea of how complex the cell is, but it still doesn't begin to answer our questions about how the cytoskeleton and how intracellular signaling is regulating shape change at below the micron scale, right? So how do we quantitatively understand how shell shape is being regulated at such a small scale, right? In general, there are many difficulties associated with understanding 3D images. I'm thinking mostly of 3D fluorescence images, right, where we have one molecule essentially lit up and the cells are moving. We have problems with big data, we have problems with visualization. All of these microscopes, for the most part, are custom microscopes, and they're designed to only be understandable with some sort of image enhancement that might vary from microscope to microscope. Cell segmentation, or in segmentation in general, is a surprisingly unsolved problem. We are still better than even a very good artificial intelligence at distinguishing objects from the background and so on. And remember also, we have the problem of our feedback loops, right? So we have cell morphology is governed by cytoskeletal dynamics, which is governed by intracellular signaling dynamics, which in turn controls cell morphology dynamics, right? Just to give an example of how complicated feedback loops can be, you can think of a pendulum. A single pendulum is extraordinarily predictable, right? It's a symbol of how, it's a symbol of the power of science, right? If I push a pendulum, it will come back. We all agree on this. But if you do something as simple as put a pendulum on a pendulum, all of a sudden we have a classic example of mathematical chaos, right? So if you add even a very, very simple feedback loop, you cannot. The characteristic of mathematical chaos is that two conditions that are very similar diverge exponentially from one another. So there's simply no way to predict, given where this pendulum starts, where it will be in a few minutes, right? Or even less than a few minutes, right? You cannot predict the fate of this pendulum. And that's unfortunate for us because the cell has many, many feedback loops, and we would like to know, given the current state of the system, given the current state of the melanoma cells, what will happen in the future, okay? And so what do we do? How do we begin? I don't really have a good answer yet, but how do we begin to make sense of all of these feedback loops? And it's not impossible. Weather prediction is the famous example. Weather is also chaotic, and we can predict the weather, right? Okay. And we also have even greater complexities at hand, which is that the cell isn't one thing mechanically. It's sort of a composite of moving goos, a composite of moving slimes, right? We want to be able to track quantitatively where parts of the cell are going so that we can tell how A relates to B, right? But the different aspects of the cell are all moving in different ways, not all of which are labeled, okay? So we don't want to think of the cell necessarily as one thing, but as a bunch of different things that are moving independently from one another, right? Right, so given all of that, we decided to start by transforming our problem from an image space, from a voxel space. So this is a single image of a cell, and we're flying through that image up and down. Right, so instead of thinking of our data as voxels, as pixels, what we're going to do is we're going to think of it as a surface. Right, we're going to try and analyze signaling and shape change from the perspective of the cell surface. Right, so here I found the surface of the cell, and I also additionally projected the fluorescence intensity onto the surface. Right, this happens to be KRAS V12, which is largely in this case just a marker of the cell membrane. Right, so regions where there's a lot of KRAS V12 show up here as red, and regions where there's less show up as light blue. Right, so now we have some sort of indication of local signaling intensity or of local anything in fluorescence, and we've put that on the cell surface, okay? And what this allows us to do is actually a little bizarre. Most analysis is done in that image, in that voxel space, but once we generate a surface, once we put data onto that surface, we're no longer in the realm of computer vision, which is the computer science field that's often applied to images. Instead, we're in the realm of computer graphics, right? Computer graphics is the field that creates Pixar movies and video games. The Pixar movies and video games represent your environment as surfaces. And here we've represented the cell as in exactly the same way. We've represented it as the same type of surface, right? And that means we can take algorithms from computer graphics and try and use them to better understand the coupling of intracellular signaling and morphology. Okay? Right, so the first thing I did with this notion of, remember, we're stealing ideas from computer graphics, is to note that here this is PI3 kinase. This is a biosensor for PI3 kinase activity on the cell surface, right? PI3 kinase activity is well known to locally grow cell, is well known to locally be associated with growing protrusions. And you see that's true here, right? Regions of red tend to grow. This is a single melanoma cell again, and regions of blue tend to retract very slowly, okay? But we don't want to try and analyze PI3 kinase activity. We don't want to try and average it over the entire surface. That would be pointless, right? We want to find regions where we can analyze it locally. And an obvious local thing that we can extract are these motifs, in this case, the blebs, right? Remember, the blebs expand and retract about every 20 seconds, right? So the first thing that I did to help make sense of these 3D images was to make a motif detector, right? And cells extend and retract many different types of protrusions. So I made a generic motif detector that can detect whatever sorts of protrusions the user is interested in, right? These are, again, lamellipodia on a dendritic cell. These are blebs on a melanoma cell, and these are filopodia on our model for a lung cancer cell, right? And we can use it in other systems as well. These are dendritic spines. And here I also clustered the spines by shape. Right? The spines are sort of this is a fluorescence microscopy image of spines, so they're sort of at the limit of resolution, but nonetheless, you can tell that the spines have different shape. Okay. In our biology-centric, I guess, methods of distinguishing signaling activity on the cell surface was useful, but we were wondering if we could instead use techniques from computer graphics to analyze the distribution of signaling, or really anything on the cell surface, in a more unbiased way, right? So can we, for example, find how PI3 kinase activity is spatially organized across scales on that cell, right? And this project was principally done by a postdoc in my postdoc lab when I was an instructor, right? So I'm the last author on the paper, and she is the first author, right? So if we ask a somewhat more broader motivating question, imagine we have many different patterns on the cell surface. We could have a punctate pattern or a patchy pattern or a polar pattern, and we want to statistically compare patterns across cells, but cells have greatly varying morphologies, right? These are all different types of cell morphologies, right? So how do we compare? How do we say that one pattern is more punctate than another on the cell surface, given that this data is on such varying domains, right? So if we imagine I were to give you a 1D signal, right? This is just a sort of oscillating structure of some sort. The very simplest thing we could do to find the spatial organization of that signal is to do a Fourier transform, right? Which means that we can decompose the wave into many waves, each of set frequency, right? So we can say that that strange oscillating structure you gave me is 0.2 of a wave at one frequency and 0.3 of a wave at another frequency, right? And the Fourier transform is extraordinarily common. Another way of thinking about that is if I had data on a sphere, I could also do essentially the same thing. This time we call it a spherical harmonic decomposition of data on the sphere, right? You may have seen these before. They're also, strangely, also just the electron orbitals, right? So if we can, just like we can do a Fourier transform on a 1D signal, you can do a spherical harmonics transform for data on a sphere. And that same math, it's honestly the same math, the abstraction of it allows you to define a set of waves on any surface, right? So here we defined a set of modes, a set of waves on the cell surface, and each cell, because it has a different shape, will give you a different set of waves, okay? So this particular bit of math is called the Laplace-Beltrami operator, and it's actually used throughout computer graphics, not because they have data on surfaces, because they largely don't, but because it's usually much faster to do computations in spectral space in Laplace-Beltrami space than it is to do computations in regular space, okay? So this is an extremely optimized operator in computer graphics, and we're going to see if we can use it to analyze biological data, right? And largely we can. There's one subtlety, which I maybe won't tell you about. We have to Well, we have to average across the different orientations of the spatial scales because we only want spatial scale. We don't care if it's if a polka dot is there or if a polka dot is there, right? And here, in order to test our system, we've taken real cells and we polka dotted them. And we expect that if we analyze the frequency of the polka dots, that this frequency should be much higher than this frequency, right? And that is indeed what we found. So these cells here out in green, the information on the surface, which remember is like our biological signal. This is at high frequency and this is at low frequency. And it's not as strange as it might sound to polka dot cells, right? If you were to test the optical properties of a microscope, we often use stripes, okay? Stripes at various distances apart. We can't stripe on a convoluted cell morphology because the stripes will run into each other. So we decided to test with polka dots, right? And what this gives you in practice is a way to ask at what scale is information on the cell surface structured, right? So here we're looking at PI3 kinase activity. PI3 kinase activity, because it's associated with the cell front, is often polarized, right? Which is a low frequency signal, right? And indeed, if we measure PI3 kinase activity on the cell surface and we inhibit that activity, we see a preferential drop in frequencies at low levels, right? Which is what you would expect, right? So our PI3 kinase inhibitor is affecting signaling only at lower frequencies, right? That is a little bit abstract, but it's from a mathematical perspective, actually pretty computationally, actually pretty conceptually simple, right? And there's one more analytical step, and then we'll go back to our biology. Okay. So the two analytical methods I showed you so far don't involve time, yet we want to know how cell signaling couples to morphology over time, right? So we decided to do essentially the simplest thing we could do, and that's track the cell surface. This has actually been done quite a bit in 2D to understand how locally cell shape dynamics couple to signaling dynamics. So here we have a 2D image of a cell edge. This is very zoomed in. This is a single cell. And this algorithm is tracking the edge of that cell. It's creating what the algorithm designers call windows. And we can peer into a particular window and track that window over time to see how the velocity at the cell edge and signaling are coupled within that window, okay? But the problem is, although this has been done in many different ways for 2D, we really want to do it in 3D because cell morphology, again, is three-dimensional, and because we can't flatten cells onto a 2D surface without changing many of their properties, right? So we want to be able to track the cell edge in 3D. And to do that, I went into the computer graphics literature and asked if we could steal some algorithms from them. And there is, it turns out, a somewhat similar problem in computer graphics called shape correspondence, right? So imagine you, say, put a pair of pants on this armadillo creature, and you want to put the same pair of pants on the armadillo creature in another frame, right? You don't want to have to draw the pair of pants again. You can use algorithms to match the pair of pants here to the pair of pants there, right? And these are in part called shape correspondence algorithms, right? But that's very similar to what we want to do. For the most part, they don't have they don't have a series of data. They just have sets of shapes, but nevertheless, it's quite similar, right? So I looked through, again, looking through the computer graphics literature to try and figure out which algorithm would be best for biology. And I found an algorithm called functional maps. So in a functional maps algorithm, instead of directly trying to map a point to a point in frame M, say, to a point in frame N, which is very hard to do, instead you paint the surfaces with functions, which are you can exactly think of as colors. Mathematically, they are simply colors. And then you associate the colors in frame M to the colors in frame N, right? So instead of directly mapping between points, all we're doing is coloring all of the surfaces and then trying to find maps where those colors best match with each other, right? And here we can remember also we were displaying biological information like PI3 kinase activity as colors on the surface. Right, so not only can we encode shape information, which is what they're doing here, but we can also encode things like PI3 kinase activity onto the cell surface and use that to generate our tracking mapping, right? So it allows us to flexibly incorporate biological constraints. And when I went into the graphics literature, I got it to work, but I wanted it to work really well. So I contacted Max Ovchannikov, who came up with this functional maps framework. He's a computer graphics professor now in France. And we worked out a way to do it in a more physical way, right? So we actually let users set conservation laws. So it's sort of as if you're imposing a physical model of how the cell moves on the cell, and then fitting that model to cell motion. Okay. Right, so this is one of our earliest trackings. This is a dendritic cell imaged 60 seconds apart. So imagine the dendritic cell at time 0 is in blue and in gold is in 60 seconds, and it's moving quite a bit, right? So even though it moved a lot, we can still generate a nice coherent mapping across all of those changes, right? And that means we can not only track cell motions locally, but we can also do things like do the calculus of signaling on the cell surface, right? So this is PI3 kinase activity, and this is the rate of change of PI3 kinase activity. So we can ask how not only PI3 kinase activity couples to motion, but how a temporal derivative of it couples to motion, or how we can do more complicated derivatives on the cell surface. Okay. Right, so now we have something of the beginnings of a framework to understand shape in 3D. We're still working on a variety of other algorithms because really, as far as I know, no one else has gone into the computer graphics algorithm and stolen it, to the computer graphics field and stolen the algorithms for cell biology, in part because we have much higher resolution data than many other groups. We were able to do that, right? So there's still a lot of low-hanging fruit there. Right, so this is our motif detection again. This is strangely our Fourier mode analysis, essentially. This is the 256th mode, right? The mathematics generates these modes on their own. You see how they conform to the surface. And this is a surface tracking of a cell with philopodia, right? We can naturally track across even waving philopodia without telling the computer anything about philopodia. We'll go back to the biology. Hopefully that wasn't too technical. Okay, back to our question of how blooming melanoma cells migrate. Now we have more of an analytical framework to begin to try and figure this out. Right, so the first question we asked is, these blebs are extremely dynamic, right? We wanted to know if perhaps the blebs were interacting with the collagen to enable these cells to get through. All right, so here we track the collagen motion. Right? These cells are in collagen ECM, and the cell surface motion only on blebs that are expanding, right? This plot might look a little weird, but it's exactly what you would expect, actually. If you push on a collagen fiber with a protruding bleb, the collagen moves. That's what we see here. That's what happens in real life. If you push on an object, it moves. Strangely, when the cell begins to move even faster, the collagen no longer moves quickly, but that's also what we would expect. Because remember, the blebs are sort of snaking their way through gaps in the collagen. So the blebs are actually moving faster than the collagen around them. This is called interdigitation. It's as if the bleb is putting its fingers into the collagen, right? And we can indeed see interdigitating blebs. Right? These are blebs that are finding their way into the collagen. But surprisingly, when we looked at retracting blebs, if blebs simply stuck their fingers into the collagen and then pulled their fingers back out, these red and blue lines should overlap, but they don't, right? And that we interpret that to mean that the retracting blebs don't simply snake back out of the collagen, but instead are pulling the collagen toward the cell surface, okay? So the cells pull the collagen towards the cell surface, and indeed, the cells end up wrapped in collagen, okay? We wondered where the collagen went. If you just pulled the collagen and it didn't go anywhere, you would just end up in a giant collagen tangle. But it turns out that the cells are macropinocytosing the collagen, which is a little weird. Macropinocytosis is a mechanism for swallowing, essentially. But yet these are collagen is solid-ish, right? But nevertheless, it seems to be consistent with macropinocytosis, right? So effectively, this is one way of measuring macropinocytosis. We color the outside of the cell with a fluorescent molecule that's large. It's just a large dextran molecule, and then that molecule is endocytosed along with the collagen. And if you put in a large enough molecule, it's considered a non-specific form of ingestion, right? So that's how we would generally think of what we would generally think of as macropinocytosis, rather than a more specific form of endocytosis. Okay? So that means essentially that these melanoma cells aren't proteolytically degrading their way through the collagen. They're instead effectively eating their way through. They're munching their way through the collagen, which is creepy, right? And we use our analytical framework to try and figure out how cell polarization is established in these systems, because they're only munching their way through, they're only eating at the cell front, right? So how do these cells remember where their cell front is? This is a classic problem in cell migration. If the cells can't remember where their cell front is, they won't go anywhere for very long. And it turns out that they remember where their cell front is in a way it's very similar to mesenchymal migration, which a little bit confused us. So cells have adhesions at the cell front that polarizes PI3 kinase, which in turn upregulates branched actin polymerization, which increases bleb size at the cell front. And this is weird because blebs canonically don't have actin in them as they expand, but nevertheless, this is what we found. And the larger blebs at the cell front remodel the collagen, which in turn affects the distribution of focal adhesions, right? Adhesions only form where the cell contacts the collagen. So if it already ate its way in the back, there are no adhesions there, right? Sort of necessarily, right? But, right, and you can analyze this in a variety of ways, consistent again with mesenchymal migration. The polarization mechanism doesn't affect where the blebs are. So here we measured the location of the blebs along the cell surface. And we asked, do they tend to be in the front? Do they tend to be in the back? And they don't have any sort of preferential localization. And likewise, if you inhibit PI3 kinase inhibition, the bleb polarization doesn't change, right? So we're inhibiting polarity. The number of blebs doesn't change, but the only thing about the blebs that changes is bleb volume, right? And this is consistent with mesenchymal migration. So cells form protrusions everywhere on their surface, but our polarization machinery makes them larger at the cell front, which enables them here. Well, on a 2D surface, it would enable them to move more at the cell front. Here, strangely, it's enabling them to eat more at the cell front, right? So I think this is more properly thought of not as a mesenchymal migration pathway, but as a macropinocytosis polarization pathway that results in persistent cell migration. Right? We have a cell signaling pathway that generates larger protrusions at the cell front, so the cell can eat more only at the cell front. Okay. Right, so in summary, I might have taken out too many slides, but I showed you that melanoma cells can migrate through soft, remodelable environments, not by changing their cell shape, as previously thought, and not even via proteolytic degradation like other cells, but by sort of chewing their way through via macropinocytosis. And I also showed you the beginnings of a variety of ways to analyze 3D cell morphologies using techniques from computer graphics, right? And I have not that much experience with in vivo pathology data. So if anyone has any thoughts on ways to use image analysis for in vivo data, that would be great. This work was largely done when I was a postdoc in Gowns's Users Lab in Texas. We're just beginning to grow my lab here. It was done in collaboration with several microscopy labs who are also in Texas, right? We have built our own microscopes depending on the problem. I'm building this same microscope at UT Southwestern. Hopefully it will be done over the summer. And also Max, our computer graphics researcher in France, has been very useful. The images are rendered in a program called Chimera X, which is a program, a software rendering engine developed for rendering molecules. So in a sense, the cells are over-rendered. It's designed to give you more cues about their 3D shape than you would get in a real physical object, but that's fairly useful in cells, right? And Tom Goddard at Chimera X has kindly changed Chimera X a bit for us so we can do this. Okay, are there questions? Took out a bunch of slides too.
Thanks, Megan, for that presentation. And yeah, that was really kind of my interest in your work was, I think it generally, I guess I found it hard back in my anatomic pathology days to detect edges and some of the problems that you've been working on. So you sort of mentioned at the end, I guess, is it at this point, are you really focused on the light sheet microscopy? Or are you also looking at have you worked with other image types?
I mean, I think for our experiments, I'm focused on the light sheet microscopy. Experimentally, I don't want to get too far afield, but I'm happy to work with any sort of fun images people have. So,
any other questions?
Yes. Hi, this is Dr. Dayton from Hematopathology. I have a question, and that is, okay, so I just let me just make sure that I understood this correctly, that you are saying that there is a localized special modification of the melanoma neoplastic cell membrane that allows for micropinocytosis or phagocytosis, effectively allowing for tunneling through collagen. Is it correct?
I mean, we found this in other cells as well. But remember, this is very soft collagen. We tried a couple of other different types of cancer cells.
Great, and that was okay. And so we have a specialized modification, a specialized localized modification of the cell membrane in certain neoplastic cells that allows for tunneling that effectively facilitates metastasis. Correct?
Yeah, I mean, it's hard for me to say if it's modified exactly, but we put it, when we put these cells in an unusual environment, this is what they do, right? It's hard to say in vivo. I would imagine they wouldn't do this, at least as frequently. And it's hard to say what they would do.
Are there any normal human cells that you know of that have this functionality?
We haven't actually, we've only looked at cancer cells, unfortunately. This is what one of the reviewers asked, but we've only looked at a few different types of cancer cells.
Because the obvious question for a pathologist is if this is a specialized modification of the cell membrane due to a specific mutation in neoplastic cells that gives them this functionality, allows them this functionality that may facilitate metastasis. And if there is some way to detect this difference in the surface membrane with an immunohistochemical stain, that would be, or heck, if we knew what the specific mutation was that allowed for this membrane alteration, to look for that by a molecular diagnostic or FISH or, other diagnostic modality. And then if there was a way to reverse engineer either the mutation or the cell surface membrane alteration that would not adversely affect any normal human cells, then you'd be curing cancer.
Well, I mean, we still don't even know how these cells are breaking the collagen. That's a sort of foundational problem. We do know that the people have shown with the same cell line that the glycocalyx on blebs can grab the collagen. Okay, so the glycocalyx is probably involved. There's some weird hypotheses that it might be someone suggested to me that the glycocalyx has acidic domains and it might be sort of dissolving the collagen. And it turns out that synthetic collagen, like in a lab, is easier to dissolve than other types of collagen. So we may have sort of amplified it. But that's the closest we have to a hypothesis.
Okay, thank you.
Thanks.
So this, just to follow up, the collagen is, how do you come about the collagen? I guess.
I mean, it's bovine collagen. It's a fairly standard procedure to put cells in this type of collagen. We buy it from companies. And it does have some unusual properties, but it's also a very standard assay. We tried making the collagen less digestible to acids, and this process still does occur, although it occurs less.
Okay, any other questions? If not, we are going to thank our guest today, Meghan. Thank you very much for joining us. Your graphic work is incredible to look at. Thank you all for joining us today.
If anyone ever needs computer graphics, let me know.